Hi, everybody. My name is Russell Laman. I'm a double major in marine science and conservation biology. And today I'm going to be showing my research using photomosaics to analyze structural complexity and conservation success of restored Belizean coral reefs. In addition to the kind of analytical side of my project, I'm going to be sharing a photojournalistic project I did where I actually traveled down to Belize and took a lot of images of the corals. So you'll see those throughout the project, throughout the presentation. Coral reefs are a really important ecosystem. They make up 25% of marine organisms are reliant on coral reefs, but they only make up 1% of the world's oceans. And so in addition to providing this immense habitat, they provide key ecosystem services such as shoreline protection and tourism. However, as we all know, corals are in peril, with 50% of corals having died in the last 30 years and 90% projected to die in the next century. And this is due to several anthropogenic forces such as coral bleaching, ocean acidification, pollution, and coral disease, like the coral you can see here on the right. In response, many conservationists have turned to coral reef restoration as a potential solution. And this consists of planting corals onto reefs, which helps rebuild the seal population and quickly can re-establish those key ecosystem services. And this is done mainly with these fast-growing branching corals, like the one you can see here on the bottom right. This is called Acropora cervicornis, or staghorn coral. And it's the main target for restoration in the Caribbean, versus something like this, which is a big brain coral and grows much more slowly. However, a few studies have analyzed the impact that planting just these fast-growing branching corals has on the reef. As a result, my honors research looks at the change over time of these restored reef sites at Laughing Bird Key, Belize, using data from 2014 to 2021, a large time series that is almost unparalleled in reef restoration analysis. My analysis was done using photomosaics, which are compilations of thousands of images that were taken at the same time on the reef and then compiled using computer software into one single high-resolution image. And these are really valuable because they allow you to take the reef back to the lab. So instead of spending hundreds of hours measuring corals in the water, you can do that on your computer at home or with a computer program, so it's much more efficient. You can also use these mosaics to make 3D models, such as this one, which can let you see really clearly how the coral cover changes over time. So as I click through the years here, you can see the amount of this scraggly branching coral really starts to increase, especially by the year 2020. And these 3D models also are really interesting because they allow you to ask much more interesting questions about the change in the 3D structure of the reef that you wouldn't be able to with just the 2D mosaics. And so going into my research questions, firstly, I asked, does coral reef restoration positively impact reef health? And how can analysis of restoration success inform future conservation decisions? As I mentioned, part of my project consisted of me actually traveling down to Belize with Kilachand support. I was able to work with Fragments of Hope, which is the restoration organization that has been planting all these corals. And I actually got a chance to snorkel and photograph these same reefs that I was using in my analysis. And this helped me gain a much greater appreciation for the reefs, but also helped me to contextualize and understand my analytical data. So getting into those, at face value, this restoration was a success. The percentage of the coral cover, specifically the coral that they were planting, did increase over time. And this was simply calculated by taking those 2D mosaics and laying hundreds of random points onto them and then just categorizing what was underneath each. And looking at, this is what it actually looks like on the reef. With every coral you can see in this image, a result of restoration. And so the whole coastline of this island is just full of these staghorn corals. The next thing I calculated was Shannon Diversity, which is a common indicator of richness and diversity of corals on the reef, with a higher value indicating a more diverse habitat. And the figure above compares Shannon Diversity to staghorn coral cover. And as you can see, there's a negative relationship where when the staghorn coral cover increased, the diversity decreased. And this actually kind of makes sense when you go and see the reefs in person, because there's some swaths like this where the staghorn coral has taken over the whole reef and spread out and turned the whole reef into a monoculture. And in some cases, has even overgrown these massive corals, like this brain coral that was already there. And so you can see this is a really unintended negative consequence of the restoration, where the restoration actually leads to decreased diversity, which is bad because the diversity of corals on the reef is really what leads to the massive diversity of species that we can see. For example, little dash gobies that live on these brain corals, to tiny crabs and Christmas tree worms, and even flocks of pelicans that are constantly circling around the reefs and diving for little fish that live in the shallows. The next major component to my analysis consisted of analyzing the structural complexity, which can be thought of as how variable the topography of the reef bottom is. You can imagine a complex reef habitat would provide the necessary niches for all the types of marine organisms versus something like a sandy bottom. And so structural complexity has commonly been used as one of the best proxies for reef health. And so a huge portion of my project actually consisted of developing the methodology for these calculations, which consisted of putting those 3D models I showed you into a 3D modeling program called Rhino, and then running a bunch of Python scripts on them. And those output some of these established measures of structural complexity, like vector dispersions, which you can see here. And this measures the small-scale reef complexity and can be thought of as the amount of habitat for the small fish on the reef. And as you can see, that is increasing over time in all the sites I analyzed. And looking at the actual reef itself, this absolutely makes sense. There's these branching corals provide a ton of habitat for these little fish, and so are essentially acting as a nursery ground with just hundreds of these little fish hiding in and amongst the coral branches. The next thing I looked at was rugosity, which can be thought of as the large-scale variations in terrain and more of the habitat for larger fish. And as you can see, there's not the same trend in the rugosity data. In fact, while there's some year-to-year variation, there's no real increase or decrease in the rugosity. And this was really surprising me, but made sense when I went down to Belize because there's some areas that were incredibly diverse with tons of big fish, like you can see here. But as I mentioned earlier, there were some areas that look like this, that were monocultures where the staghorn coral had taken over. And this is because the way staghorn corals grow is they do have some dispersed spawning, but they also have the ability to, if there's a storm, the little fragments break off and they can just grow up in between all the gaps. And so they can really take over the whole reef quite quickly. And so my trip to Belize really showed me just how important coral reef restoration is, but also indicated that clearly more analysis needs to be done to optimize the restoration efforts. So in conclusion, my analysis showed that restoration increased the amount of coral on the reef. However, increasing staghorn coral was associated with lower coral diversity. And restoration increased the habitat for small fish, but didn't really impact the amount of habitat for larger fish. Finally, I'd like to mention a few restoration suggestions that I already shared with Fragments of Hope. Such suggestions are really critical in this field because coral reef restoration is still so new. Firstly, I think a big step would be to plant the corals a little bit farther apart on the reef. For example, if you planted the corals twice as far apart, you could plant on twice as many reefs, restoring twice as many sites. And also, you could kind of mitigate this impact, or at least slow down this effect where the corals have come together and taken over everything. And so you could keep diversity high while optimizing your ecological benefit. Secondly, I think placing more of an emphasis on planting a wide diversity of species is key. It's really easy to fall into the trap of just planting the easiest, fast-growing species, but really for long-term restoration success, there needs to be a wider diversity. And finally, there needs to be an increase in the number of long-term studies on the reef on these restored sites. This is one of the longest-term restoration studies that's been going on at 7 or 8 years. And so more studies and we're just starting to see these key results. And so longer-term studies are really needed if you want to see what happens to a restored reef after 5, 10, or even 20 years in the future. I'd like to end by thanking my advisors, Les Coffin and Ethan Dial, and everybody in Belize for supporting me while I was down there. Thank you very much.
Can we have questions right away?
Are staghorn corals ever considered an invasive species in other contexts, or is it really only in this restoration context that they act like this?
I would never consider them an invasive species because they're one of the key corals in the Belizean reefs. But there are definitely some reef patches where the staghorn corals are kept in check, and there's patches of them, and there's some coral restoration sites, like sites in the wild, where they've kind of taken over, and you get a whole stand of them, and the whole reef is just those corals. So they provide. I don't want to be to say that having a big stand of these corals is a negative thing. They provide a lot of habitat for the little fish, but certainly if that's the only thing you're doing for restoration and everything turns into that, then that's a negative outcome. Yeah.
Just to make sure, these are all your photographs?
Yes.
From Belize. Gorgeous.
Thank you.
I have a follow-up question. So like for these reefs, you just said that it kind of act like a monoculture for some of the sites you visited.
Mhm.
So in this case, I guess I'm wondering, would there be like desire or effort to like reverse this, like possibly like transferring part of it to somewhere else and kind of increase the diversity, or is this like a practice that's like wouldn't be a good idea? I'm just kind of curious.
Yeah, I haven't heard of people taking the corals off the reef once they've already been restored. But I think the best solution would be to just enact different restoration practices. Restoration is still pretty new, and so it's really variable too. In this site, the corals grow super, super well, and so you kind of get this effect. Whereas in other sites, you may have a problem with like half the corals are dying or 3/4 of the corals are dying, and so you need to plant them super densely in order to get any kind of benefit. And so it's really on a case-by-case basis, but yeah, hopefully these results can help inform at least the people I was working with to improve their conservation success.
Okay, thank you.
Yeah. Yeah.
Hey, so you talked about the different growing times of corals. So you said staghorn coral grows really fast, whereas some like brain coral grows really slow. What's like the relative time frames for that? And do you think there's any kind of feasibility in like planting slower growing coral while the fast coral grows somewhere else, like stuff like that?
Yeah, so the difference is, so if you plant a brain coral that starts, let's say, like this big, in 5 years it'll be this big. So very, very slowly growing. Whereas if you plant a staghorn coral, if you plant a little branch that's like this tall, in 5 years it'll be like this big, like a full coral. So there's a pretty staggering difference in the rate of growth. And part of that is that the brain corals are much stronger and much more robust. But people are trying. There's lots of people trying to figure out that problem. I know there's some people that like they're creating these like shells where you can plant like a number of these like tiny little brain coral fragments together, like separated evenly across the whole shell, and they'll actually grow together, and to create a big coral much faster, because like when they're really small they grow faster than when they're bigger. Like they slow down as they keep growing, and so if you plant like little tiny corals like right next to each other, they'll actually grow into one bigger coral. So there's people trying to solve that problem. It definitely needs to be solved, but it's hard. It's a hard solution. It's just they just grow slower. Yeah.